Morning, everyone. Welcome to you all to this second masterclass in the area of packaging for healthcare. I am Paola Beldo from Customer Health from Emilia-Romagna region, and I'm very pleased to see you all. Just some information before starting with the masterclass. First of all, this meeting, as we said, is the second masterclass in the field of healthcare and packaging for healthcare. There will be 2 additional masterclasses that will follow this meeting. All masterclasses are open to the public and they are recorded and the presentations are shared. This meeting today, we are going to discuss sustainability and legislation for health packaging. This meeting has been organized by Cluster Health in collaboration with BalticNet Plasma Tech. And with this, I'm happy to give the floor to the co-host, Glenn Ryall from BAPNT. So Glenn, the floor is up to you to introduce our speakers and the topic of today.
Thanks, Paola, for the introduction and welcome everybody. We'd like to jump straight into our first speaker. We have Dr. Elisa Resca. She is a researcher at the Tecnopolo Mario Veronesi, if I'm saying that correctly, in Mirandola in Italy. And she brings with her extensive expertise in the area of international standards. And today she'll be presenting on navigating ISO 11607, key requirements for sterile medical device packaging. So please, Dr. Resca, take it away.
Thank you to everybody. And okay. Tell me if you can see the presentation.
Yes, we can see it clearly.
Yeah? Okay, thank you so much. So yes, thank you everybody. I'm Elisa Resca from Tecnopolo Mario Veronesi. It's located in Mirandola in the north of Italy. And today I try to speak to you about the ISO 11607 about medical device packaging. So So the talking points of today is why packaging validation is critical for patient safety and regulatory compliance, and what we can learn so we can understand the ISO about structure and purpose, recognize core requirements, and common mistake in packaging. So a few key concepts to better understand the webinar. So the sterile barrier system, also called SBS, is the minimum package that minimizes the risk of ingress of microorganisms and allows aseptic presentation of the sterile context at the point of use. The protecting packaging, that is the additional layer protecting the sterile barrier system and is content from the time under assembly until the point of use. And the packaging system is the SBS and the protect packaging altogether. So, okay. So why packaging validation is critical for patient safety and regulatory compliance? Because it ensures that medical device, pharmaceutical, and other care products remain sterile, functional, and effective throughout their intended shelf life and distribution cycle. So what's the role of the packaging in maintaining sterility? So the packaging system is what preserves the sterile state of a medical device after sterilization, during storage, transport, and handling until the point of use. It acts as a sterile barrier system, preventing microorganisms from entering and ensuring the device can be opened aseptically in the operating room. If the package design, material, and seals integrity fails, sterility is compromised, even if the sterilization process was effective. The point 2 is the risk of packaging failure. So infection, recalls, or non-conformities. If packaging fails, it can lead to serious risk such as infection due to the loss of sterility, product recalls if multiple units are affected, and non-conformities identified during audit inspection. By integrating usability engineering into packaging design, testing how user open, handle, and interpret the package, manufacturers can prevent these failures and demonstrate compliance with the MDR Annex 1 requirement. So it's the point 3. The connection with MDR requirement is called in the Annex 1, paragraph 11.2 and 23.3. In details, the paragraph 11.2 is packaging must be designed and manufactured to ensure that the device maintains safety and performance during transport. And the 23.3 is focused on the information provided on packaging and labeling. So just a brief overview of the ISO. The ISO is the global reference standard for the packaging of terminally sterilized medical devices, and it ensures that packaging system maintains sterility up to the point of use and designed and undesigned, validating and controlled appropriately. It is divided into 2 parts, the part 1 and the part 2. The part 1 is the focus of the part 1 is the design, the material and performance requirement, and the focus of the part 2 is the validation, so forming, sealing, assembling, and ensure that packaging process are designed, validated, and maintained so that every sterile barrier system reliably protects the medical device until the point of use. So this is the standard. The part we can start with the part 1. So here the main topics starting from the chapter 4. So we have the general requirement, the material, performance, sterile barrier system, and sterile barrier system in chapter 5. The design for aseptic presentation, chapter 6, the usability evaluation, chapter 7, chapter 8 and chapter 9 are about the packaging system and performance testing and validation and changes, inspection in chapter 10 and the annexes from A to F. So what is general requirement? The packaging must protect the device from contamination and damage during handling, storage, and transportation. Must be compatible with chosen sterilization method, for example, steam, heat, radiation, etc., and must be validated to maintain sterility until the expiring date. So the material required must be biologically safe, so no toxin and harmful substances, physically and chemically stable under sterilization condition, and compatible with the device and sterilization method. In chapter 5, we talk about the sterile barrier system, and is defined as the layer that maintains sterility until the device is used. So the requirements include integrity, must prevent microbial ingress under normal handling and transport, performance, must survive stress from sterilization, transport, and storage, and compatibility, must not react with the device or compromise functionality. In chapter 6, we can find the design for aseptic presentation, so the packaging system requirement. A packaging system can include the multiple layer, primary, secondary, and tertiary, and the requirement included protection for physical damage and environmental factor, so humidity, dust, UV, clear labeling, device identity, lot number, sterility information, and validation for seal strength, durability, and microbial barrier performance. So what about the primary, secondary, and tertiary layer? So just a few key terms. So the primary packaging is directly contacts the medical product. For example, blister, packs for pills, vial, syringe, and is the SBS. Secondary packaging is holds primary packaging for added protection and branding. So it's additional protection, for example, carton boxes. And the tertiary packaging is bulk packaging for shipping and distribution, so it's storage and logistic, for example, pellets. There's also the specialty packaging type that are designed for specific risk control and performance needs. So in chapter 6, we talk about the design and development for packaging system. So packaging must be designed to maintain sterility until the point of use. And all the procedures shall be established, documented, implemented, and maintained. The medical device component and construction that contributes the right fluid path closure assembly shall be identified. In chapter 7, we talk about the usability evaluation for the aseptic presentation. So to ensure that SBS and packaging design allow user to present the device aseptically, that is without compromising sterility at the point of use. So the key requirements are aseptic technique compatibility. So packages must support aseptic opening, handling, and transfer on the device to the sterility field. And the design should minimize risk of contamination during this action. The user evaluation, the usability of the packaging system must be evaluated under simulated or actual use condition. So it involves a representative user, for example, nurse, sterile processing staff, and observations should confirm that user can open and present the content without contaminating the sterile device. The design factor to evaluate is to identify the opening or peel direction, grip area size or resistance to tearing, peel force and control during opening, and the visibility of sterile and non-sterile zone, clarity of labeling, and the packaging shape and stiffness. And the last, the documentation. Result of usability must be documented and justified. So in chapter 8, there are the packaging system and performance testing. So packaging system performance testing, for example, the protection. The packaging system must protect all the SBS and contents from hazard during handling, distribution, and storage. Hazards include shock, vibration, compression, temperature, humidity, and so on. Note is adequate protection is confirmed if the sterile barrier system passes integrity testing. So the testing scope, the performance testing should be conducted on packaging system with the worst case sterile barrier system and protecting packaging. Another note that the performance testing doesn't need to be repeated when new seal equipment with the equivalent technology is introduced, provided the sealing process is validated and meets the prior specification. The worst case identification is a rationale for identifying the worst case sterile barrier system and be established and documented. Another note that is a worst case scenario consider exposure to all specified sterilization process and the most challenging content. The stability testing demonstrated that the sterile barrier system maintains integrity over time. A real-time aging, stability testing should use real-time aging methods and accelerating aging. Accelerating aging can suffice the climate expiry date until real-time aging data is available. Note that accelerating aging should commence with the 3 months of real-time aging unless an alternative rationale is provided. The real-time aging must begin before commercialization. A rationale for accelerating aging condition and test duration must be established. A prior testing. Previously documented tested is sufficient unless it's demonstrated that the contents adversely interact with the sterile barrier system. So, the sterile barrier system integrity testing must be conducted after performance and stability testing on sterilized sample, and the testing can assess the integrity of material seen closer separately. Note 1, loss of sterility is considered event-related, not time-related, and note 2, that the performance testing evaluates the interaction between the packaging system and the products under manufacturing, sterilization, handling, and storage as shipping condition. So stability testing ensures that sterile barrier system maintains integrity over time. In chapter 9, we have the packaging system validation and changing. So revalidation and change management ensure that packaging system continue to maintain sterility and comply with performance requirement throughout the its life cycle. The inspection prior to accepting presentation is in chapter 10. So we have a visual inspection. It is the best practice to visually inspect all SBS label as sterile immediately prior to use to define if any branches in integrity are evident. User instruction, when user-specific instruction for use are appropriate or required, they should include the guidance of how to visually inspect for branches of packaging integrity prior to use. In chapter 11, we have the information to provided. It is sure that the user and the regulators have all essential information to maintain sterility, handle packaging safety, and understand performance and regulatory compliance. So in September 2023, we have an amendment A1 of the ISO Part 1, and the most significant difference with respect to the 2020 version are the following. Annex ZA has been included for giving evidence of the harmonization with MDR of this standard. Annex ZB has been included for giving evidence for harmonization with IVDR of this standard, so the in vitro diagnostic, and Annex F of the normative and Annex G about the informative have been added for describe requirement about packaging risk management. So just a few points about the ISO Part 2. The scope of the Part 2 is focused on the requirement for validating the process used to form, seal, and assemble packaging system for terminally sterilized medical device. While Part 1 deals with design and performance of the packaging system, Part 2 ensures that the manufacturing process consistently produces packaging that maintains sterility. So the main topics of the Part 2 are validation for forming, sealing, and assembly in Chapter 5, so IEQ, OQ, PQ framework, routine process control and revalidation, common pitfall, assembly, used reusable barrier system, and sterile fluid packaging. The main requirement of the ISO Part 2 is a validated packaging process. All sealing and forming process must be validated to show they produce consistent and compliant result. The equipment qualification and maintenance. Packaging equipment must be qualified, calibrated, and maintained to ensure process stability and reproducibility. And the operator training and competence. So personnel performing packaging operation must be trained and competent with documented evidence of training. And where also the environmental control, that is packaging area must have controlled environmental condition that prevent contamination and variation. The process monitoring and documentation, continuous monitoring and recording of process parameters are required to demonstrate control and traceability. And the revalidation and change control. Whenever equipment, materials, or process change, revalidation must be conducted to confirm continued compliance. So also for the Part 2, in September 2023, we have an amendment. And the most significant difference with respect to the 2020 version are the following. Annex ZA that have been included for giving evidence of the harmonization with MDR. The Annex ZB that included for giving evidence that harmonization with IVDR. And Annex B for the normative has been added for describing requirement about packaging risk and management. So what the integration with the other standard? the standard, the ISO 13485 about QMS and documentation, so quality management system for medical device. The ISO 14971 about the risk management. The ISO about sterilization validation. The EHC 62366 is about usability. Usability is summative and formative for accepting opening. And the ISO 15223 part 1 that is about labeling. These are just a table about shall be recognized as such. So the symbol to recognize sterilized packaging labeling for medical device. And we have also some just the few last slides about the some common non-conformities. For example, the lack of complete validation for sealing or forming process. For example, a company manufacturer pre-sterilized a purchase for surgical instrument. They validated the sealing process only for one lot at room temperature, but the production also occurred at slightly high temperature. Later, during transport, some seal failed, compromising sterility. So this is for an example of non-conformities. Another example is the inadequate aging study or missing real data. For example, a manufacturer claims a 2-year shelf life for the sterile barrier system, but only performed accelerating aging testing and never conduct real-time storage study at room temperature. So regulators note that the actual shelf life could not be confirmed. Another example is insufficient documentation of material specification or supply control. For example, a sterile package uses a new type of medical grade film. The supplier certificate states of the material is suitable for sterile medical packaging, but the company does not keep a detailed test report or incoming inspection records. Auditors flag this as insufficient control over the critical packaging material. And the last one is the missing usability evaluation for accepting opening. For example, a sterile tray with a surgical tool is sealed in a punch. During use, nurse find the peel open feature too stiff, causing accidental contamination. The company did not perform user testing to simulate accepting opening, which is required to ensure the package can be safely opened in a clinical setting. So the best practice are develop packaging early in the device design process, perform risk-based validation and testing, document the design rationale and material compatibility, include usability and accepting handling evaluation, and maintain traceability of packaging component and process. So the main recap, the ISO ensures sterile integrity and patient safety, require design, validation, and documentation alignment, and integrates tightly with ISO 13485 and ISO 14971. Thank you.
Thank you so much for your presentation, Elisa. It was great to have a sort of in-depth look at how important and all the steps maintaining sterility of packaging and how that sort of has a downline impact. So it's really important. It's really important we have an idea of how that impacts the packaging decisions. I just wanted to mention too for the room, if people have questions, please feel free to add questions in the chat. And, you know, if people want to provide answers, they can do that as we're talking. But I want to keep moving to our next speaker as well, who I'm delighted to introduce. He's also from my hometown in Australia, Melbourne in Australia. So it's great to find another Australian over here in Europe working in this field. So our next speaker is Edward Kosior. He's the CEO, founder and managing director of NextTech Limited. It's a company gaining recognition for its innovation in plastics recycling and sustainable packaging solutions. So we're moving the step further from the regulations, now we're looking at the sustainable packaging solution and how it can impact the circularity. With Mr. Kosior's extensive expertise in advanced recycling technologies and circular economy strategies, he will present today on the innovations in the recycling of healthcare packaging. So, Edward, please, the floor is yours.
Thank you very much, Glenn, and thanks very much to Elisa for setting the scene on these packaging materials, especially with issues on sterility. So we've heard about how specific the performance of packaging has to be, but we now, if you like, are facing some new issues with all of these specifications based on performance. So first of all, what we see in the healthcare sector is that plastics are used everywhere. It's almost the sign of a modern healthcare system that we are using quite specific and in some cases quite complex delivery systems for all types of aspects of healthcare. And now one of the issues that I think we are all facing is we're all aware that plastics are being produced, but very often we don't have an end-of-life scenario in mind. And I was particularly listening to the ISO standard, waiting to hear that maybe there will be some reference that maybe we should pick environmentally friendly packaging, but it just focuses on sterility, which is completely understandable. But especially in the new amendments, maybe they'd say something like, let's pick packaging that has some positive contribution to a low carbon footprint or end of life, but it's not in that scope. So just to give you a global picture, the Healthcare Plastics Recycling Council estimates that something like 15 million tonnes of healthcare plastics are produced annually. And this produces approximately 5% of the total global plastics waste. So this is a really big contribution to the waste stream. And as you know, plastics are made in many formats, and they ideally should be recovered and reused. If we go to the parallel area of packaging that's used for consumers, we have a regulation which says that by 2030, there are targets for recycled content for packaging. For things like PET bottles, it has to be 30%. For contact-sensitive products like food-grade products, it needs to be 10% from 2030 and up to 50% by 2040. And then for other packaging, it has to be 35% by 2030 and 65% by 2040. This is an attempt to direct the business world and the consumers to understand that we need to create circularity in plastics packaging. And of course, when the packaging is being recycled, it has to come back to a food grade status, and so that it can be used back in contact with food. And that is indeed the biggest market for plastic packaging globally. When we look at single-use plastics in the healthcare system, we have a wide range of plastics being used. Here are 7: fluid bags and tubing, gloves, rigid devices like syringes and sample tubes for various purposes, device packaging like the ones we've been referring to, sterile pouches, flexible films, protective materials, PPE that health professionals wear, and also pharmaceutical packaging and wipes. All of these are very common, very widely used globally, and the materials are typically polyolefins or low-density polyethylene, polypropylene, high-density polyethylene, and PVC. And in many cases, there are complex combinations of these materials, especially in blister packs. So first of all, when we look at, if you like, the range of materials, we have quite a wide range of materials and very different formats where they're used. Most plastics generated in clinical settings, they're neither sorted nor recycled. They go straight to clinical waste, which means high temperature burning for cytotoxic waste or low temperature incineration for other medical plastics. And in some cases, the materials can be sent for autoclaving and landfill, or perhaps other applications. And for clean materials, they can be sent for mechanical recycling and, in other cases, landfill for low-risk materials. So these are the destinations. And of course, what we would probably say is very little material from clinical settings or healthcare settings are actually recycled globally. So it's not a very good picture as far as the end of life of these materials go. So perhaps we should focus on, what have the health industry have been doing in this area in trying to, if you like, reduce the level of waste that's generated, especially in a simple and safe way. And this is really important. Obviously, we're not going to relax standards of safety and sterility. They have to be kept, but perhaps many other things can be done. And indeed, much of the material that are generated in, say, a hospital setting are non-infectious, and many of these single-use materials can be recovered and recycled. But there are many which need to be treated correctly as well. So in this particular one, this is based on IV bags. There's an example of a hospital in the US that was trying to collect bags. And so in their trial, their pilot program, they collected something of the order of 170,000 IV bags, which is equivalent to 6 tons of waste. And they diverted that material into industrial products. Now, the IV bags are made from PVC, so they can go into typical flexible PVC flooring applications. And the key thing here was this wasn't a special program in the sense that it introduced anything new. It just added an element of identifying and redirecting the non-hazardous PVC bags into a new collection process. And there's another example where a Canadian hospital was concerned about the usage of rubber gloves. Of course, rubber gloves are widely used, and this is a really interesting one because in this particular case, this hospital was using 33,000 gloves per month. So you can imagine how many gloves in that hospital are being used, and then multiply that by all the hospitals. The numbers are quite staggering. It's something like 10 billion gloves are used annually. So in this case, the hospital launched a program about appropriate and inappropriate glove usage, about when gloves should be used and how they should be used. And then in 6 months, just through an education program, they reduced their glove use by just over 50%. And they saved using 90,000 gloves. And of course, this also saves CO2 emissions. And they exceeded their initial 10% reduction target. And it just shows that by looking after the environmental aspects and setting some targets, you can actually make inroads, because in many cases, people do not appreciate or worry about the end of life or the waste stream that's being created. So the other area to just briefly touch on is one that's ubiquitous across the world, which are the medication blister packs, which are very widely used for dispensing pharmaceuticals. And these materials look simple because they are designed for sterility, for single-use applications, and designed for end-use friendliness. These materials are often quite complex in terms of material composition. What you may see is as a plastic metal layer could be in fact 2 or 3 or 4 layers of plastic combined into one, and then with a metallized surface on the top, but often bound together with adhesives to make the whole thing hold together. And in one case, one of the hospitals, this case in Australia, in Bathurst, they started to introduce a program to recycle blister packs. In reality, this is a collection process because what they do is collect the materials. The next step is, what on earth do you do with these? Because they are complex to recycle, and they would probably go into various processes. And I will mention that this area of blister packs is something that we, Nextec, are working on. It's a project that's coming up very soon. It will be announced later in November. But I just mentioned that this is a challenge that we are looking at because of the complexity and the technology we have available. In another Queensland Children's Hospital, they also started to collect blister packs, and they collected in 3 months 16,000 blister packs. Not a lot of weight, but it's the concept that these materials need to be avoided from landfill, which is typically the destination, and then they have the capacity to be recycled later by whatever technology is available. So just stepping aside and thinking, okay, what would happen if we do collect materials? What are the technologies available to recycle them? And I'm listing here the techniques that are currently used. So the most common way of recycling plastics is to sort them into monoplastic streams, so that we have, if you like, only one type of plastic present, and then we can actually recycle that material in a relatively straightforward manner. And of course, the process of recycling has to, in the case of food grade packaging, has to eliminate contamination and any potential contamination. And the most common techniques are mechanical recycling, and I've listed some here. Now, what I haven't listed is pyrolysis techniques, where materials are pyrolyzed into hydrocarbons. Due to the complexity of packaging and the presence of chlorinated plastics like PVC and PVDC, things like pyrolysis are not going to be applied to hospital waste. When it goes into a waste stream, it goes into high temperature combustion, but there's no recovery of any materials. They simply get broken down to fundamental atoms. So I'm going to mention processes that dissolve plastics. These are solvolysis processes where you can recover the polymer after dissolution and recovery. And then thermal desorption processes where monopolymer streams, after sorting, can be actually heated, melted, treated under vacuum to extract contamination and bring that material back to a food-safe process. Now, this is currently not being applied to pharmaceutical plastics, but in the upcoming project that I mentioned, this will indeed be the case. And then the last one is a solvent extraction process, where you can extract the polymer with a solvent to remove potential contamination. So here's one. The first one, this is from a company in the US called PureCycle, where they are dissolving polypropylene in butane and under high pressures and relatively high temperatures, 140 Celsius. And then after a series of steps, which I won't go into, they filter the material to remove colorants like pigments. And then in a later step, they precipitate the material by pressure reduction. And the idea is that you can take any material, in this case, or any polypropylene material, and you can remove the constituent additives, including colorants, fillers, stabilizers, and end up with a material that looks like a virgin plastic and with high purity. This is a very expensive process, and it's one that's probably 4 years down the track. Another one that is rather interesting and closer to home, this is a company called Reventas in the United Kingdom, and they are taking, for example, on the left-hand side, Sharps containers, and those they are a mixture of materials but monopolymeric. And then after dissolving these materials, they can bring it back to an unpigmented resin. So this is a program that they are setting up right now, and these are the fundamental steps of feedstock preparation. In other words, making sure it's correctly sorted, dissolving the polymer in solvents, filtration of the material to remove particulates and colorants, and then finally the separation of the polymer from the solution, and then the drying and extrusion of this material back to a granule. So this is quite a novel process. It's gaining a lot of attention globally, but potentially it has the potential to be used for healthcare plastics as well. But the preliminary step of sterilization is quite important, because these materials would be hazardous otherwise and couldn't be processed in any separate facility. Just to go on to now thermal desorption processes, what I'll mention is a program that will be applied to healthcare packaging in the near future, one we call Next Loop. This is designed for recycling polypropylene, and it contains important elements of recycling which I want to show. And this is a process that creates a food-grade stream of polypropylene. In the creation of this program, we have something like 50 members all working on the circularity of food-grade packaging. And of course, when it comes to healthcare packaging, we need a similar spectrum of companies working on, if you like, the end of life of healthcare packaging materials, which will be the case. And so in the recycling of these food-grade packaging, there are two important steps. The first one is the sorting, and the second one is the decontamination step. And I just want to show you how we're using artificial intelligence to identify and separate food packaging, separated away from non-food packaging. And this is done by an imaging system where we train a identification program or a deep learning program what food-grade packaging looks like and what non-food packaging looks like. Then we can program the system to target the packaging we want to select. So in this case, we are selecting food-grade packaging that is white and natural. And this process is running at 15 tons per hour. And so you can see here, packaging going over those illuminated bands, and then they are being ejected by air jets at the end of the belt. So this allows us to separate food from non-food. The target for this process is greater than 95% food packaging, less than 5%. And this process achieves 97%. And of course, this goes further. This is the first step of a complex process. But my point here is that we can use AI sorting systems and train the AI system to separate many formats of packaging. And this will start to happen, I believe, in even the complex area of clinical waste. This is what polypropylene packaging looks like en masse. When it leaves your home and goes to a collection system, it ends up in bales and it can then be processed. So we can take that material, take it through all the sorting, cleaning, washing steps, and then we have a decontamination process at the end where we use specialized equipment. In this case, I'm showing you some of the internal parts of an extruder that has very high surface area where we can apply deep vacuum and extract any contaminating molecules from the material. And to demonstrate the decontamination efficiency, this process is tested by artificially doping in model molecules, materials called surrogates, for potential consumer contaminants. And so we have different molecules of different polarity and volatility going from low molecular weight to high molecular weight. And in this case, we can demonstrate the power of the system. So we can characterize, and then we can do migration modeling to predict whether these materials will migrate, for example, into food. And it can be done comparably to other materials like healthcare packaging. So quick illustration of the power of the system. This is headspace GCMS, gas chromatography, mass spectroscopy. These peaks are on individual flakes of polypropylene after washing. And then after the process, we see no peaks. And this is, if you like, evidence that we have extracted the materials that were previously present in the material. So it's indicative of the cleaning process. The analysis actually goes a lot further than that, but this is a nice illustration. The last process is a process for cleaning films, which is being used with supercritical carbon dioxide as a fluid for extraction of these materials. And this process actually is being used to take films that have been used by consumers and then extract them with supercritical carbon dioxide as a solvent. So under supercritical conditions, carbon dioxide is a very effective organic solvent, and it can remove oils, it can remove any flavor and fragrance molecules, and it has also the capacity to de-ink, and also in some cases can demetallize and delaminate multi-layer films. So quite a powerful system. And when we look at the decontamination rates of this system, we see something quite unique. The solvent-based process has very high decontamination efficiency, up in the 99% across the board, and it's not dependent on molecular weight, whereas the thermal processes rely heavily on time and temperature. In this case, the use of a solvent rapidly changes the process, and this is again a GCMS trace. The lower curve is film before treatment, showing peaks, and also the curve there is representative of high molecular weight waxy materials, and on top after supercritical CO2 treatment, showing no peaks and removal of the waxy materials present. So just coming to the end of this very short presentation, what has to happen in the future? Because we are at the very beginning of the consciousness of the volume and the need to treat clinical waste. And of course, it's going to start with the whole collection process, because to recycle and to recover these materials, even into an open loop, we need to collect the materials properly and safely. And of course, this means if a hospital or a group designs a program, they need to take the waste companies into, if you like, partnership, so they can design the appropriate system. And the whole system has to be focused on environmental performance and not just waste disposal, which is very often the current focus for the end of life. In other words, get it off the site and don't worry about it. In reality, we do need to think about it. The other issue is on product design. I was listening to the explanation on the standard, on the ISO standard, and there's no mention there about designing a product for eco principles. And of course, these can be and should be built into the design of each product, and even in medical waste. In other words, putting a new feature in there which promotes the sustainability of the packaging, so that it doesn't affect performance, but we think about and simplify the next step for the package by building this into the design. The other really important area comes back to regulations and standards, where they have to be set up so that we can actually direct the end of life of these materials as well. In the recycling of sharps containers, for example, they are currently mandated. They have to be disposed of by incineration. If some novel technologies turn up, which include sterilization and recycling, then we need to adjust the regulations to allow these innovations to go forward. And so eventually we need regulators in the government, but also regulators in hospitals to adopt principles of circularity in their procurement practices, and they should be looking at materials that lower the carbon footprint and lower the end of life of these materials, and so that they don't just end up as waste, and also avoiding problematic materials for given applications. So apologies for all this detail, but hopefully that's been of interest to you. Thank you.
Thank you so much, Edward. It was fascinating to see how, some of the potential ideas for innovation and how, all these little things impact the circularity, and how we need to sort of be more mindful in how we look at our legislation and how we look at our rules that can impact directly about our recyclability of items and stuff. We've got a couple of moments now for some questions. If anyone has any questions, please feel free to put your hand up and I can head to you or put it in the chat. I did while people are thinking about their question, I did have one question for you, Edward, just around if you have identified, actually, there's a question from Merce. I'll hold my question and let Merce ask hers. Merce?
Yes, thanks, Glenn. I also have a question for Edward. I also think it was really, really interesting, all your presentation. I'm going to go deeply about it when you into it when you send it. But I don't know if I didn't catch something. You explained that at the beginning, well, or at the middle of the speaker, you said something about how was it that there is that you can recycle polypropylene in a mechanical way, but it has the properties as virgin PP. Did I hear it right?
That's right.
And can you explain it a little bit more? So how is it?
So if there are two processes I mentioned which claim that they can actually take a material that has had, if you like, additives put into it. What they do is they dissolve the polymer in a solution. They take various steps to purify it by filtration, for example. So using carbon filtration to remove pigments and color. And then you end up with a solution of polymer and solvent. And then by changing your conditions, so you can get the polymer to precipitate out from that. So you end up with a fine powder. So that material, in theory, should be as the same as virgin, so clear, with absent with any additives in there. So it should be without those materials. The big question is always what residues will be left in that material. Will it be indeed equivalent? Will it need any post-processing? And this is really where this upcoming project, it's going to be announced in this month. It's called SUSPAC, which is Sustainability of Packaging in the Healthcare Sector. 13 organizations will be working on this project, and we'll be looking at combinations of processes that might end up with closing the loop. In other words, taking syringes that are in sharps packs and then recovering the material back to a virgin compound and then giving it back to the syringe manufacturers to make more syringes. In other words, having a closed loop process. It sounds like a very adventurous one because there are many hurdles. It's quite ambitious to try to close the loop. But even if we can take it out and turn it into, if you like, non-food applications, it still has a huge potential as well. So the target is to close the loop for sustainable packaging. But the idea is indeed to create a material that looks like and behaves like virgin plastic, but it's been recycled by this process called solvolysis.
Okay, many thanks. And make sure, I don't know if in the first slide it was that, but on the last one I couldn't see your mail. But for sure, it will be a good idea to get to be in contact with you.
Yeah.
I am from the packaging cluster in Catalonia, and yes, just make sure that you add that on the slides.
It is on my last slide. I'm sorry, I stopped before I got to it.
Okay.
It is on the last slide.
Then I'm going to receive that. No worries. Thanks.
Thank you, Merce. And yes, I was going to say also, Edward, please keep us in the loop around SUSPAC or where we can find information about that upcoming project, because it does sound very interesting. And I know there'll be even people that are maybe not on this call that might be interested in this further down the field. So it'd be interesting to keep aware of that. I did just have one last question as well. Like, are there particular, from your experience and also possibly Elisa, are there any particular barriers that you have seen, I guess, putting it into the EU context or, around legislation and sustainability? Have you seen any particular barriers that EU packaging industries have around, sustainability, or is it just the same kind of rules across across all, sort of sectors, whether that be EU, UK, or, sort of in a global context?
The regulation of the medical device are very, very, very strict. So there could be a lot of different SPS barriers that we can try, but for the MDR and also for FDA, we have just a few kind, because the regulation is not still so improved. So the Tyvek or the polypropylene or something similar, but all the new types of barrier need to be regulated and or validated and certificated. So it's very, very difficult for the medical device to find a new kind of packaging more compatible and recyclable. So it's very difficult. It's the medical device aim that is very strictly. Yes.
Yeah. Was there anything, Edward, like that you
Sure.
noticed through in a European context?
So the EU is risk averse. They worry about risk. And so everyone works to minimize risk. And that's why we have not touched this area of healthcare plastics for a long time. And I think now that we realize that we have a global problem with plastics waste, and we have to think in Europe, we have waste management systems. What happens to medical waste in countries where we don't have good waste management systems? Where are they ending up? We know where they end up. They end up in landfills and they get scavenged and picked up. And we even hear about syringe recovery and reuse from those materials. So quite scary scenarios coming out of it. But coming back to barriers, and take US and Europe as the scenario, the big issue is what you can do, the tightness of the regulations around medical waste. If you like, the lack of openness to innovation at the moment. I think this is really where our project is wanting to go, to experiment with different approaches to create sustainable materials. In the area of operation of hospitals, some of those examples highlighted that just by educating the hospital staff and operations, you can actually redirect a lot of material away from hazardous waste into conventional recyclable materials. So some good hospitals are already doing this, and they're doing this because their waste bills are very high and they want to reduce their waste bills. To give you an example, something like the high temperature waste materials that have to be incinerated, they cost £1,000 per ton to dispose of. Whereas if you like, the non-hazardous materials, £100 a ton. So a huge difference between the two. So hospitals can save if they take care to educate their staff, create the infrastructure for appropriate collection, and then not have, if you like, the risk of cross-contamination. So staff training is a big thing, education is a big thing, but the other big limitation is, if you like, the lack of design for recycling in the healthcare area. They're not designed for recycling, and everything's been thrown into, if you like, the difficult basket, or shall we say, the performance basket. If we started to design for recycling, we would do many things. Quick example is the use of PVC in IV bags, blood bags, and tubing. We consistently use this material for its properties, but PVC has to be plasticized. Plasticizers can leach into the liquid products, and PVC is a technically challenging material because it dehydrochlorinates when it's processed, and that creates hydrochloric acid, which damages processing equipment. So in the packaging sector, everyone's moved out of PVC. The healthcare sector strongly sits in that area, making, if you like, a difficult to recycle material. So we really need the manufacturers to take the message on. They need to design for recycling. And this is one of the big limitations. People are sticking with what they know because it's safe. But is it optimum? The answer is no. We need to change the thinking. We need to change the design. We need to change the products. We need to do the research. We need to prove we can go to a more circular economy and reduce waste. So I'm sure the future is going to be a very busy one. Even though it's difficult, it's one that we can make a lot of progress.
Thank you so much for your time, Edward and Elisa. We're sort of running out of time here at the moment, and we really appreciate sort of, the insight into these challenges and things that we need to sort of think about moving forward. So I really do appreciate your time. Also, for the people that are on the call or for further distribution, we will have this session available on our YouTube page. An email will be going out to all participants with all that information shortly. So thanks again, everyone. And we look forward to seeing you in our next session. Thank you. Bye.
Thank you very
Thank
much,
you.
Glenn, and thank you, everyone.
Bye.
Thank you. Bye-bye.
Bye. Bye.